Yeah, thank you very much. It's a real pleasure to be here today. I'm going to try to make up some time, so I hope that's going to work out. So my talk is structured into three main topics, and the first one is the research part, and then the second part, the role of patient organizations, and then just very quickly I'm going to touch on something that's a new idea of TESS, which refers more to plans for the future. So I don't think I have to say much about this. As we heard before from Phil, cilia occur all around the body in humans, and there's mainly two groups, motile cilia that move, which when there's a defect in genes encoding for proteins in these cilia, you get primary ciliary dyskinesia, whereas most cells throughout the body have non-moving cilia, just one cilium per cell, and this gives rise to more complex developmental disorders like Bardet-Biedl syndrome, Alstom syndrome, Jeune syndrome, Joubert, and all these diseases. So the common thing that links all these diseases is that it's caused by mutations, so-called mutations, which are kind of spelling mistakes in the recipe how to make ciliary proteins. And if we imagine that all the genetic information in the human body could be compared to a library, then it would be a spelling mistake in one little book, one letter is wrong in one book in the entire library. So it's a really, really tiny mistake that leads to quite severe consequences. And probably you've seen that many times as well. Most of the ciliopathies are recessively inherited. This means that you need two faulty copies, two copies of the gene that has the spelling mistake, one from mom and one from dad, in order to have the disease. What is special about the ciliopathies, as Phil said before, there are some symptoms that occur in more than one of the diseases. So for example, extra fingers or kidney disease can occur in Bardet-Biedl syndrome, in nephronophthisis, in Jeune syndrome, in lots of disorders. But not only that, also we have many genes found to be carrying spelling mistakes in ciliopathies, and spelling mistakes in the same gene can give rise to more than one disease. So that makes the picture very complicated. And together there's far over 50 genes known to cause ciliopathies, so that's a very large number. And if we think of this library, then if you look at all these books, somewhere in one of them there's one letter wrong. But where will it be and how are you going to find it? So this was the big problem in the past, that this was very difficult and took just far too much time to find. So from a research point of view, this really has changed since we have new sequencing techniques that are called next generation sequencing techniques. And compared to before, where we had to open every single book and read it from beginning to end in the entire library to find the spelling mistake one after the other, we can now kind of open all the books at the same time. And if you imagine maybe on your computer the search function, you just put into search something you search for, and in theory, you're going to find it like this. So that in theory is much faster, and also in practice. I mean, it's not 100% working for everyone, but it is much faster. So that's really a huge progress, and I think that will revolutionize the diagnostics in ciliopathy. So if we look at what we've done like in London, so we've looked at a number of different diseases with different amount of patients, but we found that the vast majority of BBS patients here, shown in blue, are the patients where we do know the genetic defect. You can see there's only very few patients where we do not know. And this is patients investigated with the new method. So this is not a high number of patients, but that works really well. And for other diseases like Jeune syndrome here, the numbers are not as great, but it's like probably 2/3 of all patients where we do know the genetic cause. So overall, all the ciliopathies together with the new technique, probably we can solve at least 2 out of 3 patients. So that's much better than it used to be. So why do I think that's important? It's not only because if you know the name of your gene, that's somehow making you more happy. That's probably not the case, but there are some things related to the genetic and the phenotypic consequences. And in some diseases like Jeune, this is just an example, there's a high phenotype variability. Some people have very short ribs, which gives them really strong respiratory problems. Some people have longer ribs. They don't have the same extent of problems in BBS. Some people have early eye disease. Some people get later eye disease. And it's not understood why this is. And one reason it could be is that this is a genetic reason. So with the new techniques where we can see all the genes, we hopefully will be able to make more conclusions. And for Jeune syndrome, this is just because this is mainly the disease I'm working on. And we found two things, that if you have mutations, in a gene called DYNC2H1, then you have very short ribs, which gives you very strong respiratory problems, but very rarely you have kidney or eye disease. Whereas if you have mutations in a different gene, usually you don't have very bad respiratory problems, but you pretty much always have renal failure in childhood and retinal disease. So it is helpful in some cases to know the gene, because that helps you to make predictions about the disease course, which then in turn you can maybe tailor your clinical care and also the planning of the future around that. So as I said, this has implications for clinical management, but also sometimes it helps us to make the actual diagnosis. So we had one case which we thought is polycystic kidney disease, because clinically the baby had this and nothing else, but genetically this was a case of BBS, which I think is important for the parents to know, because this means the disease course will be quite different. And we had several cases of Jeune syndrome who in fact have a completely different disease, absolutely unrelated. It's not ciliary, but it comes with the same skeleton phenotype. And this implicates a very different treatment for these patients. So sometimes I think it is important to find the correct diagnosis, because this really changes the treatment. And without genetics, we would have not made this diagnosis. So what is the benefits of knowing what gene mutation you carry? It's good for you, yourself, for your individual therapy, but also maybe for the entire family, because it enables prenatal diagnosis and carrier testing. And maybe even more important, is important to understand why these spelling mistakes lead to the disease. And that takes me on to the next part. In research in ciliopathies, many people try to solve the question, how does this spelling mistake here in the DNA give rise to all these problems? So one thing that we often use in research is patient cells, as like I think someone from Alstrom group mentioned before, that we like to take skin cells and then look at these cells, and we can look at the actual cilium, like here seen in green and red, and we can see, for example, how long that is. So in that example, this is a Jeune patient on this side here. The cilia are a bit longer than in the control. We also have cases where they may be really short and stumpy, so we can check, is the cilia looking normal? Is anything wrong with it from outside? For PCD, we take samples from the nose where we see how the cilia move. Are the cilia okay? We can also check what Phil said earlier, because many of the proteins are involved in transport to build the cilium. So we can check, is there any accumulation of things that should be transported, or are there things that do not reach the tip? So for example, here, the control patient, you can see this molecule here is very weak, but in the patient it kind of accumulates in big blobs at the tip because it cannot come back from the tip. So that kind of tells us something is wrong with the transport from the tip down back to the base. And the third thing we check for all diseases, if we have a patient mutation, sometimes the protein just doesn't go to the right place in the cell. So in this example, here in the control, this is the protein where it should be. It's at the base of the cilium. So the cilium you can't see because it hasn't been visualized. But here, these two mutations, you see there's nothing here. So with these two mutations, this protein does not go to the base of the cilium anymore. And that kind of helps us maybe to understand how this disease occurs. So if we don't have patient cells, what we can do as well is that we take other cells and we artificially get rid of the gene. So there are methods to disturb the gene, which kind of mimics a patient if you want. And this cell line loses the gene of interest, and we can see what's the consequences of that. For example, is there shorter cilia, or also what Phil said before, because cilia are involved in many signaling pathways, is there any defect in these pathways? So that's a very useful tool. And then maybe the most important, and that's something maybe not everyone likes, but it is the fact that for most research, we do need animal models, because in the end, if you have a cell line in a dish, this does not reflect an entire organism. So in the end, we do need animal models. And this comes from, it kind of ranges from things like green algae, which we have seen before, worms, goes over fish, frog, and maybe the most important model is mouse. So we quite like to work on fish for several reasons, because they're easy to keep. They live in like aquarium. This is in our own lab in London. And they're quite fast model. They're quite inexpensive, and you don't feel as bad working with fish as with mouse, to be honest. So that's really nice, because we do not work with these adult fish here, but we only work with embryos. So the fish lays eggs, and that sounds a bit weird, but we put some marbles into a plastic box and put them into the tank here. And in the morning when the light comes on, the fish lay eggs into these marbles. And then we just put them in this noodle sieve. And we inject something to block the gene, then put it into an incubator, which is just a warm cupboard where the eggs grow for 5 days. And that's it. So this is very simple, and it does not involve any kind of adult fish. So this is very nice. You can see here in green, these are some cilia in the fish. This is the fish embryo. So this fish is maybe like 3 days old. And here we got rid of a gene that encodes for cilia protein. And you can see the difference. The tail of the fish is really curved, although his cilia look pretty normal. So that is nice to kind of get rid of a gene and see if this gives rise to a phenotype compatible with ciliary function. Also, we can test specific patient mutations when we are not sure if this particular mutation causes a phenotype. We can test those. But there's a problem. As you can see, the fish does not really look like a human, and it has a curved tail that has not really an obvious relationship to the human phenotype. So then sometimes there's no way around it. Then we do need a mouse model, because mouse is much closer to human, and some things, like for example the weight gain in ciliopathies or the extra fingers, are obviously more similar. So for some reason we still do need a mouse, but we do try to start off with fish. So for therapy research, I think there's 2 different therapy options. So we can treat the actual cause of the disease, like the ciliary dysfunction, or we can work on making this supportive or symptomatic therapy better. And I'm only going to talk about the causes therapy today. And I think there are 2 major areas. And one is genetic therapy, where we try to correct the gene, or we try to correct the pathway downstream of the gene. And for gene therapy, there has been one paper, which was shortly mentioned by Phil as well, where a mouse that cannot smell, which has a ciliary defect, was treated with gene therapy. And they actually were able to make that mouse smell again. So in mouse, that already worked. However, this is not a human therapy at the moment, but there's hope that at some point that will develop into that. Also, there's therapies with a drug. Some mutations make a protein really short. So it's shorter than normal. And there's a drug that, for that particular type of mutation, can kind of go over that position where it tells to stop the protein and make the protein the normal length. And that has been tried for other things in animals and seems to work. So there's no reason why that wouldn't work in ciliopathies. However, that is not at the stage to be tested in humans at the moment. But again, there's hope that at some point this will develop into it. And then the last thing would be to test drugs that affect pathways that are downstream of the cilium. So for that, we in London use fish and treat those fish with different drugs and see if we can change the phenotype with that. And that currently we do for renal cysts, and there's some trial going on for retinal degeneration. But as you know, this is only a fish, so from there probably we would have to move into a mouse first, and from the mouse move into the human. So this is still quite a way to go. So in total, there's lots of research going on in ciliopathy around the world, and there's one big European consortium, if you want, which is called Cilia, which in the program has a component of applying the research to human health. However, this is not maybe to the extent one would wish. So this Cilia consortium is currently not involving any patient organizations, and I think that's one of the important things today, that there is really an important function for patient organization in order to distribute the knowledge, because the people who have the disease are the ones who know best what is important for that disease. And I think, as said before, because the diseases are so rare, only together this will represent a significant amount of people and will be enough to initiate change. So as an example, for other diseases, there's neuromuscular disease. I don't know if any one of you knows this, for example, Duchenne muscular dystrophy, but there are also other diseases, and they all affect the muscle. So in a way, this is a bit like ciliary disease, where we say everything affects the cilium. And they all came together, and they formed one big consortium, which is called Treat Neuromuscular Disease, Treat NMD. And they have this motto here, where they say they want to advance diagnosis and the treatment for those living with neuromuscular disease. And they managed to really form a very big society, and they have lots of money, so they fund hundreds of projects each year. And I think that's maybe in future, what we should kind of aim for, to become as big as that. So that's from their website. So they kind of involved into biobanking and research, so they make aims for research, but they also do clinical trials, and they measure the outcome of projects. So they involved into everything. So that kind of leads me to the last point, that's the idea of tests. So the start maybe would be to make one database where we aim for people from around the world to be able to put their contact details so that they can contact each other, but also that there's kind of a collection of contact details in order for other people to distribute information, for example, about meetings, or maybe it's about a new treatment that might be available, or things like that. So that would be a plan for the future that hasn't been started, but that would be something where we would need everyone to participate in that, because at the moment everyone can maybe contact people with the same disease within their own patient group, but it's not really possible to contact people from other patient groups. And there are obviously overlaps, like people with retinal degeneration and Jones syndrome. Maybe they would like to contact people from the retinal patient group or vice versa. So I think there would be a use of that. So yeah, so in summary, I think that genetic diagnosis is very important. The research is currently happening worldwide, but there's maybe not enough patient involvement into that. And yes, I think that patient organizations really are crucial for the advancement of the disease understanding and the clinical treatment. And I think this registry test plans or the contact database is the very first step to form one big organization for that. So yeah, thanks.